Hello and welcome everyone. In this podcast, I want to talk a little bit about how chloroplasts and mitochondria are inherited, and more importantly, how are their genomes inherited. This is a genetics class, and we've spent most of the class talking about the nuclear genes, and that's appropriate because we know more about them, and they do a lot of cool things, and now we know all that stuff. But I think it's important to talk about the other genes that are also found in eukaryotic cells. I don't want them to feel bad like we're ignoring them. So let's talk about chloroplasts and mitochondria, and in particularly their genomes. Alright, I think it's appropriate now that before we proceed that we talk about this endosymbiosis theory. And this is the theory that explains how eukaryotic cells were able to establish a chloroplast and a mitochondrion into their cells. We'll talk about the chloroplast first. We believe, with considerable evidence, that there was a cyanobacteria that could undergo photosynthesis. And then there was this primordial eukaryotic cell. Primordial because it didn't have all the adaptations that our eukaryotic cells have today. It wasn't able to undergo respiration or photosynthesis. It was still developing and still adapting to the pressures that it was encountered with. It, of course, still had a nucleus, and this primordial cell took up this cyanobacteria. Now, this primordial cell with this cyanobacterium had a distinct advantage over cells without the cyanobacteria. It could undergo photosynthesis. It was able to use energy from the sunlight. This was a very distinct advantage. Having this allowed this cell, this primordial eukaryotic cell, to evolve and adapt and become what we now know as our algae, or some of our algae, and plants. So our photosynthetic algae were derived this way. They have the chloroplasts in them as well as the plants. And so let's just draw one of these cells here. Still has the nucleus and has this chloroplast that at one time was the cyanobacteria. Over the course of evolution, genes from the chloroplast, some of the genes from the chloroplast genome, have moved into the nucleus. Because remember, the cyanobacteria had its own genome, and when it came in here, it still maintained that genome. And over time, some of those genes have been transferred, while maintaining some of its own genomes. Now let's look at mitochondria. Mitochondria came from this, what we'll call a purple bacterium. It also was taken up by one of these primordial eukaryotic cells, and this gave this primordial cell with this purple bacterium also a distinct advantage because it could now make more energy through respiration in an environment that was increasingly more and more oxygen rich. Being able to use the oxygen instead of the oxygen killing the cells was a pretty heavy advantage. So this would then become all kinds of eukaryotes, and it would eventually become the mitochondria that we now know today. And the mitochondria shares many features of what we might see with the purple bacterium, as does the chloroplast through the cyanobacteria. And just like we saw with the chloroplast, genes from the mitochondria have moved into the nucleus too, while maintaining a certain subset. Now I want to move on and just say a couple things about the genomes. Alright, let's compare the chloroplast genome with the mitochondrial genome. And I just want you to know these few pieces of information. The book says a little bit more, but this is all I want you to know. The number of the number of chromosomes in chloroplast, depending on the species, there may be between 60 to 300 chromosomes. Remember, there are going to be these circular chromosomes. They're all going to code the same genes, but there's going to be 60 to 300 of them. Mitochondria have far fewer, 5 to 10 chromosomes per cell. Number of genes per chromosome. Number of genes per chromosome in the chloroplast is roughly, say, 100 to 120 genes. Eukaryotes, depending on the species, between 13 and 20. Next, let's talk about how they are inherited. Usually, when we look at chloroplasts or mitochondria, genomes are inherited in such a way that they either come from the paternal source or the maternal source. Chloroplasts, if you're talking about an angiosperm, angiosperms are the fruit-bearing plants, and their inheritance is maternal. Whereas the gymnosperms, like the pine trees, their source is of a paternal origin. Usually. There are always exceptions, of course. Now mitochondria, these vary a little bit too. Mammals, it's maternal. We see some exceptions in other kinds of eukaryotes where it's paternal, but usually we will follow a maternal inheritance. However, again with angiosperms and gymnosperms, we see a difference. Angiosperms, just like with the chloroplasts, their mitochondria are inherited maternally as well. But the gymnosperms are paternal. I'm not sure if I pointed this out on the last whiteboard, but it's important to remember that plants did receive the chloroplast from the cyanobacterium, but they also received mitochondria, so they have both. But please do remember that plants have both chloroplasts and mitochondria. Alright, now I'm going to erase this, and I want to talk about chloroplasts first. Chloroplasts, as I said on the last whiteboard, are inherited through a maternal source for angiosperms and usually a paternal source for gymnosperms. I'm going to look at an angiosperm example here, talking about what are known as 4 o'clock plants. Usually a plant leaf, at least in this plant here, is going to have a solid pattern. Though we know there are patterns where the plant is what we say variegated, where we have some green and some not green areas. And this has been linked back to a chloroplast mutation. And the reason we have this difference in colors here is that there's more chlorophyll here, and that's what gives this a green color. In this area here where we have the white pattern here shown on the board, there's no chlorophyll there. And we know now that the reason we have this variegated phenotype is because of the chloroplast genome. So I'm going to draw a zygote here, a fertilized egg in the plant. And I'm going to draw a nucleus here, and then we're going to ignore it, because it has nothing to do with this. I'm not going to draw the mitochondria, but they are here. I just want to focus on the chloroplast. And the chloroplast contains, as I said, about 120 genes. Many of those genes are responsible for photosynthesis. And some of those genes are important in making the chlorophyll. So I'm going to draw the chlorophylls as plus signs here. And that's because in the genomes here of these chloroplasts, the gene that makes the pigment chlorophyll is not mutated. And that's why you're going to get this plant leaf here. However, if they were all mutated, let's mutate them all now. I'm going to put minuses for the chloroplasts, because they're all mutated. If that's the case, you're going to get a leaf that looks like this. However, what often happens is a condition known as heteroplasmy. In the condition of heteroplasmy, some of these are wild type, not mutated, and some of them are mutated. And as the cell divides, what will happen is it will produce some cells with more or less wild types ones. So as this cell divides, I need to draw it up here. Let's say over time we produce a whole bunch of different cells. And because these minus and plus genomes are mixed up here, just by random chance, you could get some that had more pluses than minuses, or you could have some with more minuses than pluses. You may have some that are all minus. You may have some that are all pluses. You're going to have a difference here. That's why we call it hetero. And because of that, within the pattern of the leaf here, you're going to have regions that are, this leaf here I should have pointed to, areas that have a lot of chlorophyll, so a lot of green color. And you're going to have areas that with no chlorophyll, perhaps this cell here. Though I didn't draw it here, you're going to have some regions here that are lighter green, somewhere in between these two. That might come from this one or this one. So we get a different severity of the effect depending on how the genomes are arranged in the cells that are produced. So this is due to heteroplasmy. Now the same thing happens in humans or other placental mammals. So mitochondria. So during fertilization we have our egg here, and I'm just going to draw the mitochondria like so. Despite popular belief, the mitochondria is tubular shaped and not oval shaped, and so it might look something like that. The egg, or rather, I'm sorry, the sperm will come along, and the head of the sperm has no mitochondria in it. But within the tail of the sperm, that's where the mitochondria is. That's what gives it its power for movement. And it's not really along the whole tail, it's really only along the first third of it, so let me make the tail a little longer. When fertilization occurs, you end up with your egg here, with the mitochondria, and the head of the sperm popping in here, and the tail breaks off. Within the head of the sperm, that's where the nucleus is. And the two nuclei fuse, the oocyte and the sperm nucleus fuse to produce this one big nucleus. Now, just like we saw with the chloroplast, we can have genomes within this mitochondria that are wild type, mutated. So I'm not going to draw out all the possibilities, but you can imagine now when this fertilized egg begins to divide, you will have some cells. You're going to have some cells with more wild type, so I'm just going to put up arrow plus. You will have some with more mutated ones. You'll have some that are pretty much evenly mixed between plus and minus for the mitochondrial mutations. And if there happens to be a mutation on one of these genes that's important for respiration, you could see how you could have variable effects. Some of these cells will go on to make a lot of energy, some won't. Even the oocyte, I didn't mention this before, but the oocyte that's selected for this can also be variable. Sometimes you can have an oocyte that's all wild-type mitochondria, and then you can have some that are half and half, and then you can have some oocytes that are selected during oogenesis that only have minuses. And when we talked about Simon Keller in the case a while back, the reason some of his siblings had variable phenotypes from the mitochondrial disease that was running in the family is because some of them received an oocyte that was equally distributed, so they had mild phenotypes. Some received ones that had mainly mutated ones, and those were the ones that had major effects. So again, this is all due to our friend heteroplasmy. Heteroplasmy is what allows variable phenotypes to come from the same mutation. And the only reason this works with mitochondrial chloroplasts is because they're not diploid like the nucleus. They're multiploid. They have many, many copies of the genes. And because of that, you can't make predictions the way we would with the Mendelian ratios. Okay, well that ends this podcast. If you have any questions at all, please let me know, and I'll be happy to answer any questions you might have. I'll see you in class. Bye.